Welcome to the first in a series of videos covering FRQs from previous years. So first we're going to start with, of course, 2025, which was last year's FRQs, and we're going to start with question 1. To try and keep these videos a little shorter, I'm going to go ahead and I am going to do one video per question. So I am going to put up the first part of the question. Go ahead and pause the video at this point so that you can read it. I'm not going to waste our time reading it out loud. And after you pause it, then you can restart so we can go over part A. All right, so you should have paused it. And let's look at part A. Part A is asking you to describe the function of ribosomes. So if it's described, you definitely need to be writing in a complete sentence. Unlike an identify, where sometimes you can get away with writing just a few words or a phrase. So what do ribosomes do? Well, ribosomes create proteins. Ribosomes are functioning in protein synthesis, so you can do any of those. So you can say ribosomes. When I was doing this, I did this question without looking at the scoring guide, thought since it's a describe, I'm going to put a little more detail in there. So I wrote something along the lines of ribosomes use an mRNA transcript to produce a protein. And I wanted to include a little more detail because as long as you don't contradict yourself, you're not going to lose points for having some extra information. I am going to say when you're plotting out your questions, you're going to have about 25 minutes to complete the first two questions, the long questions, number 1 and number 2. There's not going to be anything in the timer that's going to tell you that. It's actually in the directions it's going to tell you. You're recommended to take about 25 minutes for question 1. So what I would do is I would sit there and I would plan. You're not going to get separate scratch paper, but you can write in your exam booklet. And so kind of plot out what you're going to write. So you could just write when you're planning this protein synthesis or making proteins, whatever you want to do. And then when you come back, you figure out how to write in a complete sentence and then save yourself a little space. Like, for example, this is in an exam booklet that's not separated out by part. It's just separated out by question. So you could leave maybe that much space. And if you have extra time, you could add more detail after that. So if we go through and we look at the scoring guide has multiple options for what you could put. Now, notice I probably put more detail than you needed to, but like I said, a little better to be safe than sorry. So some of the examples that are going to earn points are ribosomes synthesize proteins or polypeptides. You could say ribosomes perform translation, or that they are the sites of this polypeptide or protein synthesis. Wherever you see in the scoring guide a slash, that means either of those options work. And when you see something like this where it says examples of the acceptable responses include the following, you don't have to have all 3 of these. Any of these 3 would earn credit. So that is a pretty easy describe question. And then for question number 1, they give you data and they give you a long explanation of an experiment and some background information. Go ahead and pause so you can read this. And while you guys are pausing and reading this, I am also going to pause and I'm going to highlight I'm going to go ahead and describe why I'm doing this. So we're talking about when proteins are going to be transported into the ER because they need to be in the ER so they can be exported. So we're saying that they're either translated during translation or after translation. And then what they talk about is membrane proteins in the ER membrane that allow these proteins to enter. So it's saying that SR is necessary for transport during translation. And then it says that SEC, so I'm going to say SEC22, is necessary for transport after translation. So we have during translation and after translation. And then we have the description of an experiment. So they say they made siRNAs. Now, if you don't know what siRNAs are, because we didn't get a chance to explicitly cover them in class, it says they reduce expression. So these, whatever they are, and they're small interfering RNAs. Their mechanism is they are going to basically block mRNAs from getting translated, and so they are using these siRNAs to reduce the expression of either SR or of Sec22. And then they're using that to try and see which of these proteins is involved in transporting which of the proteins that are being produced during translation. So they treated groups with either the SR siRNA, so this is, you can think of it as anti-SR, if that makes it easier in your head, and anti-Sec62 siRNA. And to determine the relative amounts of those proteins in each group compared with cells treated with the control siRNA. So this right here is just showing you that for these control siRNAs, when they put them in, this is our relative amount of Sec62 protein and SR protein that's produced. It's not telling us how much total, it's just saying we're going to compare this to our control. So we're setting our control at 100%, and then if we look at our graph, when we add that Sec62 siRNA, which is supposed to prevent the translation of Sec62 protein, then you're going to see that only about 25% as much protein is produced for Sec22, but the SR protein actually has a larger production. It has more of the SR protein produced than with the control. So now we need to identify the dependent variable in the experiment. Remember, our dependent variable is pretty much whatever we're measuring, and that's going to be on our y-axis. So this is actually relatively easy to say. It's our relative protein content. And then when I was doing this myself, just to be on the safe side, I made sure to write either percent of control or compared to control siRNA. Sorry, control siRNA. All right, let's take a look at the scoring guide and see what the scoring guide says. And it's just saying the relative amount of protein or the amount of protein. Anything in parentheses in the scoring guide is optional. So if you don't have it, it's not going to affect your score. All right, now let's look at B part 2. So I didn't show you the entire FRQ at once, but this one has, A has 1 part, B has 3 parts, C also has 3 parts, and D has 2 parts. All right, so for B, go ahead and pause and read. I left the explanation paragraph up here as well, just so that way you guys can take a look. I also, okay, so you can pause. Hopefully you've restarted by now. And this is a justify question, which means we're going to be giving a little bit more explanation. So why they included the control of measuring the relative amounts of both the Sec62 and the SR proteins. And that's from the data shown in figure 1. If you need to go back in the video to one of the previous slides and look at the graph, you can. But it's not really necessary. You can just say something along the lines of, so when I was doing this on my own, I wrote something along the lines of using the measurement of Sec62 and SR proteins shows that treating with Sec62 siRNA doesn't reduce the expression of other proteins like SR. So that's what I wrote. Let's take a look at the scoring guide. So the scoring guide had a couple of possible acceptable responses. They're saying that this control lets you know whether the Sec62 siRNA affected the content of both proteins, or you could say it determines whether the Sec62 siRNA reduced only Sec62 or whether it reduced something like the SR protein as well. So either of those are appropriate answers. It's more important just to know that this is saying that you're looking at the fact that adding that siRNA only affects the concentration of the protein that you're looking at. And then notice we have, these optionals within the parentheses. We also have some potential alternate phrasing like reduced or affected, reduced or affected. All right. Now let's look at B part 3, which is going to be our last one with figure 1. And since we are looking at both at the data again, I went ahead and re-included the graph. So pause and read the question. All right, hopefully you have done so. All right, and now we are looking at the effect on SR protein production when cells are treated with the Sec62 siRNA. So we are looking at this, the Sec62 siRNA, and we're looking at the SR protein. And remember, we're going to compare this to the control. That's what all of this is. So we have what looks like 175% of the protein produced when the Sec62 siRNA is added. So we can say something like the production of SR protein increased. And that should be enough to earn us the credit. But if we want to be, 100% sure we're getting the credit, we can add some more detail and say that it increased to 175% of control expression. Notice since it's a description, we've got to write a complete sentence. It helps to add a little more detail. Whereas if this was just a identify, we could have honestly probably put it increased. But I like to be more specific because it increases the chance that you'll earn the point. All right, so what we're saying is that it increased, the SR protein production increased. We could also say that it increased by 65%. Whatever you wanted to say. Oh, I looked at this at an angle and it looked like 175% was what it increased to. My bad, but luckily with these graphs, since you don't have these grid lines to help you with exact measurements, they will accept a range. So if you put anywhere from it increased by 50% to it increased by up to 80%, you would still earn the credit. Now, if you put it increased by 10%, or if you put something like it increased by 150%, then you would not earn the credit. All right, so now we're looking at part C, and part C has now taken us to a different experiment. And in this experiment, now we're measuring how much has been transported into the ER of the cells. So go ahead and pause this and read the question, look at the new graph, and read C part 1. So now we're looking, hopefully you've restarted after pausing and reading, we're looking at the production of 3 different proteins. Well, not the production, we're looking at how much has been transported to the ER. So what percentage of the control is actually inside of the ER? And we're now asked to identify the independent variable. This is a little more complex than your traditional identify an independent variable, just because we have a couple of independent variables now. We still have the independent variable of the type of siRNA. But we have also added an extra independent variable of the which protein is measured. And so luckily for this, if we look at the scoring guide, they will accept either of these. It's either the type of siRNA or which siRNA was added, or the type of protein whose amount was measured. So there were two possible answers because there were two independent variables. All right, go ahead and pause and read C part 2. All right, now we are looking at an identified question, which means we don't even have to really have a full sentence. Although if you want to be on the safe side, I would always write in a complete sentence no matter what. All right, we're looking at the proteins that when they're treated with the Sec62 siRNA, so we're looking at this one, and this one, and this one. Which ones show an increase in percent compared with the control? So when you compare with the control, protein 1 is not increasing. Protein 2 is increasing, as well as protein 3. So when I answered this on my own before the scoring guide, I went ahead and put proteins 2 and 3. If we look at the scoring guide, it tells you you could have actually, since it gave you the option to list one protein or more than one protein, apparently on this one you could have earned full credit for either answering protein 2, protein 3, or correctly putting it was both protein 2 and protein 3. I will tell you, I have seen instances on the scoring where you would have had to put both protein 2 and protein 3 to earn the point. So if there is more than one correct answer, it doesn't hurt you to put it. If it was something like the previous question where they ask you only for one independent variable, they would only grade the first response you put. But since here they gave you the option to put either multiple proteins or one protein, you could have gone with either way. What I'm going to say is if you're putting more than one answer, always start with the one you're most confident with, because if they only ask you for one response, they'll only grade one response. If they ask you for 2 examples, then they'll only grade the first 2 examples. So if you write 5 things down and the 5th one is technically correct, you're not going to get the point because they're only going to grade you from the first 2. All right. Go ahead and pause and read C part 3. Hopefully you have read it. Now we're asked to calculate something. And they're telling us that we have these 2 proteins. They're telling us how many nucleotides are found in each of the genes, but they're assuming that all of these proteins encode amino acids. So we don't have to worry about whether the AUG methionine is included or worry about the stop codon. So what they're really testing you on is, do you know that there are 3 nucleotides per amino acid? So what you'll do if they want the difference in the number of amino acids is you'll take the number of nucleotides and you're going to divide it by 3. So this is for protein 2. And then we're going to subtract from that 234 divided by 3, which is for protein 1. If you've got the time, you can pause and you can do the calculations. This turns into 165 minus 78, and the final answer is 87. And I would just put 87 amino acids because sometimes you don't get credit if you don't have a unit, because they'll be like 87 once. All right, if we look at the scoring guide, that is the answer. In the brackets, they show you the calculation. If it's possible for you to write down your setup for the calculation, it's always helpful because a lot of times they want you to have the setup, and sometimes that will affect whether or not you earn the points. All right, so now we're going to D part 1. Go ahead and pause and read. Hopefully you had a chance to read it. Now we are supporting a claim made by the researchers. And so we don't have to say whether the claim is correct or not, or whether it's supported by the data or not, because we're saying it's supported. But if you want to, you can say the researchers' claim is supported because in the data, or in figure 2, the only protein, my bad, the only protein whose expression or whose concentration amount in the ER is reduced after treatment with Sec62 siRNA is protein 1, because this is the one that we're looking at. Because this implies or means that only protein 1 is transported post-translation, because Sec62 transports proteins after translation. My answers can sometimes be a little wordy because I am always worried about not quite getting credit, but let's go ahead and let's look at the scoring guide. So it's saying that only protein 1 showed a reduced amount or percentage of transport when the cells were treated with the Sec62 siRNA. Notice, remember, I always say include a little more information just to make sure that you are, earning the point. All right, go ahead and pause and read D part 2. So D part 2 was a little tough for me as well. I seem to have misinterpreted it when I read it. So it's saying that the researchers are claiming that the amino acids near the amino terminus determine how likely it is for the protein to pass through the protein channel in the ER membrane. And they want a justification of that based on your understanding of what factors will affect the transport of proteins. So this means that this protein is going to have to go through the channel. So I was thinking that about the size. So you cannot do diagrams on the AP test and earn credit, but this is just how I think. So I'm just going to do a quick diagram. If this is a protein sitting in the cell membrane, my thought was it has to do with the size. So if you want a protein, and my thought was, oh, they're trying to think of the amine terminus versus the other terminus. Well, the amine terminus is the start of the protein. So my bad, I accidentally went back. I was thinking about size, but what they were really thinking about when I looked at the scoring guide was what's going to affect what can pass through the channel. So the channel is probably going to have charges or things like that based on whatever amino acids are facing the inside. So let's say the internal charges were all positives. You want negatively charged amino acids on the N terminus to allow them to pass through, because if they were positively charged, that would block the transport because they would be repelled from each other. So saying something like that would be the amino acids. I decided halfway through my explanation that I should not go over what I thought because it would just be confusing. The amino acids near the amino terminus should have the opposite charge that's what we're going to pretend like that's an R charge of the amino acids facing the inside of the protein channel. And that would allow those amino acids to pass through because of the matching charges. If we look at the scoring guide, it says that amino acids at the amine terminus should have similar polarity or opposite charges to the protein channel, or to the amino acids inside the protein channel. That's what this section is talking about, are more likely to pass through. And notice that is enough to earn credit. You don't have to have the rest, just because that's optional since it's in the parentheses. All right, I know that was long, but keep in mind, we've spent about 25 minutes on this question, and that's about the amount of time you should be spending on this question on the AP test. Hopefully I'll be able to get up some more videos soon of questions 2 through 6 for 2025.